Now let's discuss next question, that is question number 8. We have to differentiate between repetitive DNA, satellite DNA, and then we have to differentiate between mRNA and tRNA. Then last we have to differentiate between template strand and coding strand. So let's start with the very first thing, that is differentiating between repetitive and satellite DNA. What is repetitive DNA? The non-coding sequence on DNA, the non-coding sequence which is present on DNA and repeated several times in the genome of an individual is termed as repetitive DNA. Whereas the satellite DNA is one of the various categories of repeated DNA which contains highly repetitive sequences. Now, depending upon the number of repeats, satellite DNAs are grouped into various categories such as mini satellites, microsatellites, and mini variant satellites. So this is the difference between repetitive DNA and satellite DNA. Repetitive DNA is nothing but non-coding sequences present on DNA which are repeated several times in the genome of an individual. And satellite DNA is one among them which is containing highly repeated units or highly repeated bases. Then come towards the differences between mRNA and tRNA. So mRNA is the messenger RNA. mRNA specifies amino acid on the polypeptide. mRNA specifies amino acid on polypeptides. Thus, it is transferring genetic material from DNA to ribosome, whereas tRNA, which is also known as transfer RNA, reads the genetic information which is present in the triplet on mRNA. tRNA reads the genetic information which is present in the triplet, which is present in the form of triplets on mRNA, and transfers the appropriate amino acid from the amino acid pool of cytoplasm to the growing polypeptide chain during protein synthesis. So this is the difference between mRNA and tRNA. The last one is we have to differentiate between coding strand and template strand. We know that DNA is a double-stranded molecule. The strand of DNA which is participating in the process of formation of RNA, that means in the process of transcription, that strand of DNA which is participating in the transcription process is known as template strand, whereas the another one which is similar in the base sequence of mRNA and is not coding for any RNA is termed as coding strand. So let's write the answer of 8. First, we will differentiate between repetitive DNA and satellite DNA. So the non-coding sequence of DNA which is repeated several times in the genome of an individual, in the genome of an individual is repetitive DNA, whereas the satellite DNA is one of the repetitive DNA which contains highly repeats. Now, depending upon the number of repeats, depending upon the number of repeats, satellite DNA is grouped. Satellite DNA is grouped into various categories such as mini satellite, microsatellite, and mini variant satellite. Now if we write the differences between mRNA and tRNA, so we will write the difference between mRNA. mRNA is the messenger RNA, which specify the amino acids on a polypeptide, That means, thus, it transfers the genetic material from DNA to ribosome, whereas tRNA, that means transfer RNA, reads the genetic information on genetic information which is present in the form of triplets on mRNA. Right? So this is the difference between mRNA and tRNA. Then last is the difference between template strand and coding strand. So we'll write the difference between template strand and coding strand. Template strand is the strand of DNA which participates in the transcription process that is formation of mRNA from DNA, whereas the coding strand is the strand which do not codes for anything, and is similar to mRNA except for uracil. Alright, so these are the differences between template strand and coding strand, and the differences between mRNA and tRNA, and the difference between repetitive and satellite DNA. With this, we have completed the solution of question number 8.